In the last two videos, I've looked at choosing a sample size to meet either a precision target or a sample or a power target rather. And in this video, I'm going to show you how to use some specific power tools in R that make this a little bit easier to do in practice. So this package here, PWR, contains some of the functions that we'll use here. And the first example I'm going to show you kind of goes with the jumping spider experiment that we talked about in the last couple of videos. So let's suppose, in the last video we figured out sample size that would be needed to detect a preference of 0.7 with a power of 0.8. And it turns out that someone has already thought about this particular problem and figured out a function called pwr.p.test that does this calculation. So the input to this function uses an argument labeled h, and this is known as Cohen's effect size. Actually, let's look at the help function so we can see what's going on here. So es.h. So this is an effect size calculation for proportions, and it tells us the arguments are the first proportion and the second proportion. So in this case, the first proportion is the value of p in the null hypothesis, and then the second proportion, p2, is the preference that we are wanting to detect in our experiment. So that sets up this effect size that we can then use as an input to this other function. So pwr.p.test. We can see that the first argument here, h, is the effect size. And then the other arguments, if we set the power, then it'll return, among other things, the sample size that we'll need. So let's see. So what have I done? I've done h. So let's do this piece of code now. And then let's look at the resulting sample size. So it tells us that we would need a sample size, we would round this up to 29, so we would need a sample size of at least 29 to be able to detect a preference of 0.75, so that we would achieve a power of 0.8. So we could do this for a bunch of different preferences. So 0.6, 0.7, 0.8, 0.9. So 0.7, this was the one that we did in the last video, and we figured out we needed a sample size of, what was it, 50, I think. But let's see what this particular power.p.test function comes up with. So let's run that, and then these are the results we get. So if we're trying to detect a preference of 0.6, we need a pretty big sample to do that. 193, we would round that up to 194. And so that's not surprising, because 0.6 isn't very different to 0.5, so it's very hard to detect such a small difference. You need an awful lot of data to detect such a small difference. If we're trying to detect a preference of 0.7, we would need a sample size of rounding that up to 47. And so that kind of lines up with what we found in the last video when we did this kind of longhand, and we figured out that we would need a sample size of 50. So this function is telling us 47 will do it. And then we can see as the preference gets stronger, the sample size needed goes down. And if p really was 0.9, we don't need an awful lot of data to be able to detect that kind of difference, because that's a very big difference from 0.5, and so it's going to be very easy for us to detect that kind of preference. We only need a sample of 10 to do that. Okay, so that's figuring out the sample size needed for a proportion test, binomial test. Okay, let's move on and think about a different kind of experiment. So let's think of an experiment where we've got birds laying eggs in their nests, and in this particular experiment the researcher removed a couple of eggs from some of the nests, and so when that happened, the females that were attending the nests would lay another egg. So but those kind of circumstances create an increased likelihood that the eggs would be, or rather the birds would be infected with malaria. So I guess there's stress involved with losing an egg, having to lay another one, and then you become more likely to get infected with malaria. I don't know the biology in this, I'm just guessing, but that's what seems to have happened, because in this experiment there were 15 out of 30 birds that were infected with malaria. That these were the birds that had the eggs removed from their nests, and we also had 35 control birds who their nests weren't manipulated, and only 7 out of those 35 were infected with malaria. So let's imagine that we were going to repeat that experiment on, let's say, a different species of bird. What are the chances of detecting an effect, and what kind of sample size should we plan? So what we could do is randomly sample some data. Okay, so that's what this is. We're randomly sampling data in the two groups, and then I'm going to set up a data frame. I'll show you what the data frame looks like. So in this randomly generated data, our first bird didn't get malaria and didn't have its nest manipulated by 2 of the eggs being removed. That was the same for the first 6 birds, actually. The 7th bird did get malaria, but it didn't have its nest manipulated. Okay, and so on. So all the way down. So these are all the controls, and then the 31st bird got malaria, and that was one of the birds that had its nest manipulated. Okay, and the next one had its nest manipulated but did not get malaria, and so on. Okay, so this is a randomly generated sample from this experimental situation. We could create a contingency table from that data set. Okay, so if we were trying to detect whether there was dependence between the treatment variable and the response variable, we would do a chi-squared test here, a chi-squared independence test. And so in this case, we've set the data up so that there is a dependency between the treatment and response variables, because we set the proportion of birds in the which group is this? This is the control group. So in the control group, we are saying that the proportion of birds who get infected with malaria is only 0.2, whereas in the treatment group, so the treatment group is the group that has the nests manipulated by having the eggs removed. In the treatment group, we're saying the proportion of birds getting malaria is 0.5. Okay, so we've built dependence into this experimental situation, and we've ended up with this contingency table. And so if we were to do a chi-squared test on this contingency table, we would expect to get a small p-value to allow us to reject the null hypothesis that the two variables are independent. Okay, so where are we now? We could do this a whole bunch of times and see that we get different results each time. So that's what that is. Okay, but the question is, what sample size do we need so that we could achieve 80% power in this experimental setup? Okay, so we want to be able to reject the null hypothesis at least 80% of the time. So what kind of sample size would we need to do that? Okay, so what we can do is use this power function here, power.chi-squared.test. Okay, and to do that, same kind of setup as before. We could look up the details of the test here, so we know what all the arguments are. But essentially, we need to figure out an effect size and then plug that into the function there. Okay, so let's run all this code and see what we get. Okay, so it looks like if we round that up, we would need a sample of 80 to be able to achieve 80% power in that experimental setup. Okay, finally, let's look at a two-sample t-test scenario. Okay, so in this case, I'm going to randomly sample 20 measurements from a normal distribution with a mean of 10 and a variance of 10, which will be the control group, and then 20 measurements from a normal distribution with a mean of 12 and a variance of 10, and that'll be the treatment group. Okay, so that's what I'm going to do here, if I can get my mouse working. Okay, so I've sampled my data, and then I'm going to put my data into a data frame, and then we could take a look at what's going on. So we've got the control group and then the treatment group, and in the control group the mean was only 10, and in the treatment group the mean was 12. So what kind of power is that design? Does that design have? So we can use this power function here, power.t.test, and we figure out it's only got a power of about 50%. Okay, so not super powerful, and that's because the sample sizes are just too small. So what did we have? We had sample sizes in each group, 20 in each group. Okay, so we can use the same function, and if we state the power, then it'll tell us what sample size we need. So that tells us that we need a sample size, and this is the sample size in each group. Okay, so we started with just 20 in each group, but this result here is telling us that we need a sample size of 40 in each group to be able to achieve a power of 0.8. So for this two-sample t-test scenario. Okay, so that's an illustration of how to use some of the inbuilt power functions in R to do sample size calculations.